Gene editing sounds like something out of science fiction, a way to rewrite the very code of life. But in reality, it's happening right now thanks to a revolutionary tool called CRISPR. This technology has the potential to cure genetic diseases, revolutionize medicine, and even reshape humanity's future. But it also raises profound ethical questions. This video explores how CRISPR works, what it can do, and the difficult choices it presents. What is CRISPR? CRISPR, short for clustered, regularly interspaced short palindromic repeats, is a tool that allows scientists to edit DNA with unprecedented precision. It's often compared to a pair of molecular scissors that can cut DNA at a specific location. But how does it work? The key lies in a protein called Cas9, which acts as the scissors, and a piece of RNA that guides it to the exact spot in the DNA that needs editing. This system was originally discovered in bacteria, where it functions as a defense mechanism against viruses. Scientists figured out how to harness this natural process to target and edit genes in virtually any organism. How CRISPR can cure diseases. Imagine being able to cure diseases like cystic fibrosis, sickle cell anemia, or even certain types of cancer by simply correcting a faulty gene. This is the promise of CRISPR. Researchers are already using it in clinical trials to treat genetic disorders. For example, in the case of sickle cell anemia, scientists can use CRISPR to fix the defective gene responsible for the disease. Blood cells with a corrected gene can then be reintroduced into the patient's body, potentially offering a lifelong cure. In 2020, a woman named Victoria Gray became the first person in the US to be successfully treated for sickle cell disease using CRISPR, showing the world that this technology isn't just theoretical, it's real. The potential beyond genetic diseases. CRISPR's potential doesn't stop at curing genetic diseases. It could also be used for the following: eradicate infectious diseases. Scientists are working on using CRISPR to combat viruses like HIV and even the flu by targeting and destroying the viral DNA within infected cells. Edit embryos to prevent genetic disorders. While highly controversial, CRISPR could theoretically be used to eliminate hereditary diseases before a person is even born. Enhance crops and livestock. CRISPR is being used in agriculture to create more resilient crops and animals, potentially addressing global food shortages. The ethical dilemma. Where do we draw the line? With great power comes great responsibility. CRISPR raises serious ethical questions. 1. Designer babies. One of the biggest concerns is the possibility of using CRISPR to create designer babies with specific traits like height, intelligence, or eye color. Should we allow parents to choose the genetic makeup of their children? 2. Unintended consequences. Editing genes is complex, and there's always the risk of unintended mutations. Could we accidentally create new diseases or ecological problems? 3. Accessibility and equity. Who gets access to these life-changing treatments? If CRISPR cures are expensive, will they only be available to the wealthy, widening the gap between the rich and the poor? In 2018, a Chinese scientist named Jian Kuwait claimed to have edited the genes of twin girls to make them resistant to HIV. His actions were widely condemned, and he was sentenced to prison. But the incident highlighted how easy it could be for rogue scientists to push the boundaries of ethics. The role of regulation. The scientific community agrees that strict regulations are necessary to ensure CRISPR is used responsibly. Many countries have already banned gene editing in human embryos, but the global nature of science makes enforcement challenging. It's a delicate balance between advancing scientific discovery and protecting ethical boundaries. The future of CRISPR. Despite the controversies, the future of CRISPR is incredibly exciting. Scientists envision a world where genetic diseases are a thing of the past, where cancer can be stopped before it starts, and where pandemics can be controlled with genetic tools. But it will require careful oversight, international cooperation, and public dialogue to ensure that we use this technology wisely. In conclusion, CRISPR is one of the most powerful scientific advancements of our time. It has the potential to change medicine, agriculture, and even human evolution. But with this power comes a responsibility to ask hard questions and make thoughtful decisions. As we stand on the brink of a genetic revolution, it's up to all of us, scientists, policymakers, and everyday people, to decide how we will shape the future of gene editing. Will CRISPR be a tool for healing and progress, or will it open doors we're not ready to walk through? The future is in our hands. Watch more content in science, philosophy, and psychology on Knowledge tab. Stay curious. Stay inspired.